Chapter 4, Biology of the Cell, Part 4. So, inside the nucleus, as we know, is DNA. DNA is the genetic code that codes what makes us each unique and also what makes us similar. DNA is made up of two long chains of nucleotides. So again, the monomers for DNA is nucleotides. The nucleotide, as we know, is made up of a phosphate group, a deoxyribose sugar, since this is DNA, and a nitrogenous base. And so here's the double helix that DNA forms. We can see the nitrogenous bases forming hydrogen bonds between each other, sort of the steps of the ladder, as it were. So again, two complementary strands of DNA forming this double helix by hydrogen bonding, and specific bases always pair up with each other. So G's always with C's, C's with G's, A's with T's, T's with A's, so that if you have an A on one strand, you know you must have a T on the other strand. DNA holds our genetic material, which is in the form of genes. DNA doesn't hang around loosely. It is actually packaged with proteins. This is important because if the DNA wasn't packaged, it would act like a big bunch of thread and get all knotted up and unusable. So the DNA is packaged around proteins. These are called histones. So DNA wraps around the histones, forming nucleosomes. The histone protein complexes then coil up to form what's called chromatin. So the relaxed form of DNA in our cells is chromatin. And then when it's time to cell divide, the chromatin will form a much denser packaged structure called the chromosomes. So technically, chromosomes only are around when cells are dividing. So genes, there are about 30,000 genes or so in the human genome. And the genes code for specific proteins. So each gene leads to a specific polypeptide. And so they are the books that have the recipes for the proteins that make us and let our cells function. Every gene has a beginning, which is the promoter region. This is where the start of the gene is. And it has an end. It has a terminal region, which is the stop or end of the gene. So gene goes from the start to the end. Promoter to the terminator. All right, transcription is the making of RNA, specifically a messenger RNA using DNA as a template. So messenger RNA is made to make a RNA copy of a specific gene. The DNA stays in the nucleus and the ribosomes are out in the cytoplasm, so we need that messenger RNA to get built in the nucleus and exit into the cytoplasm. So required structures, the players needed for transcription. We have DNA, of course, itself is the template. We have the ribonucleotides, the monomers to make the RNA molecule, and the RNA polymerase. RNA polymerase is the big protein complex that produces the messenger RNA. And we need, again, complementary base pairing. In this case, it's going to be RNA nucleotides to DNA nucleotides. So RNase A goes with DNase T. RNase U, or uracil, goes with DNase A. And then G's to C, C's to G's. So RNA has Q's, DNA has T's. So to start off the transcription process, the DNA is unwound at the promoter region by the RNA polymerase. RNA polymerase will attach, and then it will find the lovely promoter and get started in production of the messenger RNA. And it uses one strand only to make the messenger RNA. During the elongation process, the ribosome will move, the RNA polymerase will move along the length of the DNA. And as it does so, it will bring in a RNA nucleotide to bind to the DNA, complementary base pairing, and then attach it to the growing length of messenger RNA. Polymerase is catalyzing, speeding up this production of messenger RNA, leading to phosphodiester bonds between the nucleotide that complementary base paired and the growing RNA chain. And it will continue this until it gets to the terminator region, where the RNA polymerase will terminate, will separate from the DNA and release the messenger RNA. However, this messenger RNA isn't quite ready to leave the nucleus, so it's often called pre-messenger RNA, pre-messenger RNA. And of course, the DNA reforms its double helix. So, here, if you're given the code for DNA, A, G, C, G, T, A, C, and asked to write out the complementary base pairing in the RNA, it would be U, C, G, C, A, U, G. All right, after the pre-mRNA is formed, it gets modified. So this is important to turn it into mature messenger RNA that can be used by the ribosomes. So the first thing that happens is splicing. Turns out there are sections called introns within the pre-mRNA that are not going to be used. These introns are not going to be used, so they are removed. They are going to stay in the nucleus because they are not needed. And that just leaves the exons being the portions of the RNA that will form the mature RNA. And so the introns are removed, the exons are attached to each other, and this is called splicing. And this is catalyzed by a huge complex of proteins and RNA that's called the spliceosome. So the spliceosome does the splicing, removing the introns, attaching exons to each other. On top of this, the spliceosome could make different kinds of messenger RNA, including only a subset of the exons. So instead of having a messenger RNA with exons 1, 2, 3, 4, might make one with only exons 1, 2, 3, and another with 1, 3, 4, so that you get more diversity in the possible proteins being produced. Other changes to the pre-RNA include a 5' cap. This is a guanine RNA nucleotide being added to the 5' end, the beginning of the messenger RNA. It's capped because it's put on backwards. And then at the 3' end of the messenger RNA, a long poly A tail is added. So lots and lots of adenine nucleotides are added to the 3' end. And then this would be the mature messenger RNA, 5' cap, only the required exons, and then the poly A tail at the 3' end. So the mature messenger RNA will then leave the nucleus through the nuclear pore, go into the cytoplasm, where it will try to find ribosomes to become allow for the production of proteins. So the production of proteins is called translation. So transcription is the production of messenger RNA. Translation is the production, the synthesis of proteins. Needs, of course, the ribosome. It needs the messenger RNA because the messenger RNA has the recipe, the sequence needed. And then there are other things that are important, including tRNAs, amino acids, and so forth. So we're using the code of the nucleotide sequence of the messenger RNA to be translated into the amino acid code of proteins. That's why it's translation, translating nucleotide sequence into amino acid sequence. So, important structures needed. Well, I went over them already, kind of. You need the ribosome. You need the messenger RNA that has the code. You need amino acids. And amino acids are brought to the ribosome on tRNA, transfer RNAs. So the messenger RNA, again, linear sequence of nucleotides, carries the instructions, the code for making the proteins. And to read the messenger RNA, you have to read it in groups of three nucleotide groups, codes for one amino acid. And those three nucleotide groups are called codons. So here we have AUG, this is the codon, the code for a specific amino acid. So there's a start codon, which is also AUG, that begins protein synthesis. That's what the ribosome looks like to get the process started. Then there's lots and lots of codons after that, each coding for different amino acids. And then finally a stop codon tells the ribosome it's done, it's made the protein, it can leave the messenger RNA. Transfer RNAs are interestingly shaped single-stranded RNAs. They shape vaguely like a T. They bring in the amino acids to the ribosome. And the way they bring in the correct amino acids is the tRNA with a specific anticodon always carries a specific nucleotide. The anticodon needs to complementary base pair with the codon. So here, this codon is UUC. You look at the tRNA, it's anticodon A AG, proper complementary base pairing. So now it brings in the correct amino acid because of this complementary base pairing of the codon in the messenger RNA to the anticodon in the tRNA. Here is actually the breakdown of the code, the genetic code. There are 3 nucleotides that have to make a codon, and the codons are then used to make the amino acids. You can see there's about 20 amino acids. You can see that multiple codons can code for the same amino acid. So we got 2 examples for phenylalanine. We got 6 possibilities for leucine. You don't need to learn all the names of the amino acids. You don't need to learn about these specific codes. Just know that there is this lovely chart that will allow you to convert messenger RNA into amino acids and also stop codons for stopping the process. All right, translation. The initial phase is when the complex is formed. The ribosome forms on the messenger RNA. Ribosome then finds the start codon, this should be codon, in the messenger RNA, and then it'll bring in the tRNA that complementary base pairs with that codon, AUG, and that tRNA is always going to be carrying the amino acid methionine, which may be removed later on or not. Maybe it's needed for the protein. And at this point, the tRNA is in the P site. We have these 3 sites, E, P, and A. Elongation. The ribosome moves down 3 nucleotides. It now has GAA in its A site. So tRNA comes in that has the anticodon, that complementary base pairs with the codon, bringing a specific amino acid. Then we will have a peptide bond forming between these 2 amino acids. So the amino acids will form a peptide bond connecting them covalently. And then the ribosome will move down 3 nucleotides to get to the next codon. The first tRNA will end up in the E area, which is the exit, and will leave the complex. The second one moves to the P site, and then a new tRNA arrives in the A site with its anticodon, complementary base pairing with the codon. And this is how it goes. The ribosome moving down 3 nucleotides at a time, bringing in a new tRNA, forming a covalent bond between the amino acid and the growing peptide chain, and getting rid of an old tRNA. And that happens until it gets to a stop codon. Once the ribosome gets to a stop codon, boom, a release factor comes in, and the ribosome breaks up and leaves the messenger RNA, and the new protein is free to go where it needs to go. DNA is the control center because it directs the synthesis of proteins. It decides when proteins are made, it decides which proteins are made, and proteins engage in the majority of functions and processes in a cell. So that means the DNA controls the functions of the cell. In addition to this, the DNA has indirect control over other metabolic changes. Because of the fact it's controlling the making of proteins, this includes enzymes, and the enzymes are going to affect decomposition and synthesis chemical reactions and what is being produced. So it indirectly, by making enzymes, controls what the enzymes produce, steroids or ATP through glucose oxidation. So again, nucleus, where you find the DNA, leads to transcription, forming the messenger RNA. Messenger RNA is modified and then leaves, and then through translation, the ribosome gives us the protein. That is it for this part of life.